Dear learners, now in this section we shall study trypsin inhibitor, which is the first plant protease inhibitor isolated from soybean. The first plant protease inhibitor was isolated from soybean and characterized in 1947. Soybean trypsin inhibitor has a molecular weight of 22,000 and includes 2 disulfide bridges and is primarily an inhibitor of trypsin but also weakly inhibits chymotrypsin. It is inactivated by heat and by gastric juice. The predominant type of inhibitor in legume seeds is Bowman-Burke trypsin. It has a molecular weight of about 8,000 with a high content of cysteine, forming 7 disulfide bridges. A trypsin inhibitor is a protein and a type of serine protease inhibitors known as SERPIN that reduces the biological activity of trypsin by controlling the activation and catalytic reaction of proteins. Trypsin is an enzyme involved in the breakdown of many different proteins primarily as part of digestion in humans and other animals such as monogastrics and young ruminants. When trypsin inhibitor is consumed, it acts as an irreversible and competitive substrate. It competes with proteins to bind to trypsin and therefore renders it unavailable to bind with proteins for the digestion process. As a result, protease inhibitors that interfere with digestion activity have an anti-nutritional effect. Therefore, trypsin inhibitor is considered as anti-nutritional factor or ANF. Additionally, trypsin inhibitor partially interferes with chymotrypsin function. Trypsin and chymotrypsin inhibitors form inactive complexes with trypsin and chymotrypsin in the small intestine, thus inhibiting the hydrolysis and bioavailability of dietary proteins. Their presence stimulates an increased pancreatic activity, which may eventually cause pancreatic enlargement followed by pancreatitis. Now let us see proteases of the gastrointestinal tract. Proteases are heavily present both in the lumen and deeply into the tissues. Pancreatic proteases like trypsins, chymotrypsins, elastases, etc. are released into the lumen of the upper gastrointestinal tract where they exert digestive functions. The microbiota also constitutes an important source of proteases. Bacteria, yeasts and helminths potentially present in the intestinal lumen produce and release proteases. Serine, cysteine, aspartic and metalloproteases are expressed and released by the microbiota. Other resident cells of the intestinal mucosa produce and release proteases. For instance, the major protein content of mucosal mast cells is proteases. Mast cells release different forms of proteases like tryptase and chymase for the vast majority and also cathepsin G and granzyme B. Resident macrophages also produce and or release different forms of proteases, matrix, metalloproteinases, caspase, cathepsins L and D. So, these are the proteases of gastrointestinal tract. Now, let us study protease inhibitors of gastrointestinal tract. Protease inhibitors have co-evolved with proteases in order to control their destructive nature. Natural endogenous protease inhibitors are particularly present in the gastrointestinal tract. They are either circulating inhibitors produced at distance from the gastrointestinal tract, mostly in the liver, or are produced on the site by intestinal epithelial cells or infiltrated inflammatory cells. Serpins A1, A3, A4, E1 and C1 are circulating protease inhibitors inhibiting serine proteases such as trypsins, chymes, tryptase, elastases, kallikreins and cathepsins G. Secretory leukocyte protease inhibitors, that is SLPI, and elefin are produced in situ by intestinal epithelial cells or leukocytes. Both inhibit elastase and proteinase 3, while secretory leukocyte protease inhibitors also inhibits trypsin, chymotrypsin, cathepsin, G-tryptase and chymes. Tissue inhibitors of metalloproteinases, that is TIMPs, are ubiquitously produced as TIMP1, TIMP2, TIMP3 and TIMP4 are present in GI tract, where they inhibit a number of different matrix metalloproteinases. The caspase 9 inhibitor, which is a cellular inhibitor of apoptosis protein 2, is also ubiquitously produced by cells present in intestinal tissues. Now, what are the sources of protease inhibitors? Crops such as beans, potatoes, barley, squash, millet, wheat, buckwheat, groundnut, chickpea, pigeon pea, corn and pineapple have been identified as good sources of protease inhibitors. It has been estimated that between 5 to 10% of soluble proteins in barley, wheat and rye grains are protease inhibitors. The protease inhibitor content of such foods has a significant influence on human health disorders, particularly in the regions where people mostly depend on these kinds of foods. These natural protease inhibitors vary in concentration, protease specificity, heat stability and sometimes several protease inhibitors may be present in the same species or tissue. Now, effects of processing on protease inhibitors. A study showed that among different methods, frying was the most effective method of eliminating enzyme inhibitors from tubers. When cooked by boiling, partial retention about 30 to 60% of enzyme inhibitory activity was observed in certain tubers. Further reduction in inhibitory activity was observed in baked or pressure cooked tubers. The study indicated that processing decreased the inhibitor content significantly and the reduction of inhibitory activity was brought about by heat activation. With this, we have completed the second section of protease inhibitors. Thank you.